Bismillah Rahman Rahim. Hi everyone. I welcome you all to our YouTube channel Viral Insights. Today I'm going to discuss our article which was published last year in the Journal of Medical Virology, and the title is The Genomic Characterization of the First Mpox and Varicella-Zoster Co-infection in Pakistan through Next Generation Sequencing. In 2022, a multi-country outbreak of Mpox occurred, and the first case was reported in London, and the patient had a travel history of Nigeria. Following that, a number of cases were reported from the same city. However, none of them had any travel history from any of the Mpox endemic countries. This basically led to a widespread outbreak affecting many countries in Europe as well as North and South America. Accordingly, WHO on July 23, 2022, declared this outbreak as a public health emergency of international concern, realizing the fact that it required an international coordinated response. As of May 16, 2023, around 87,000 Mpox cases were reported, and 140 deaths were also reported from 111 countries. The most of the cases were reported from USA, Brazil, Spain, France, and Colombia. And if we look at the epi curve, so you can see that majority of the cases they were reported in August, and these 3 months, July, August, September, and even October, they were the most of the cases were reported during this time period in 2022. So three of the regions that were mainly affected were the European region, South America, and North America. Interestingly, most of these cases, and around 96% of the cases, were male, and majority of them were in men who had sex with men. And 52% of these cases were also co-infected with HIV. We have seen co-infection of VZV with some other viruses, but co-infection with VZV is normally rare and uncommon. The 2022-23 outbreak was basically due to the clade 2B of this virus, and the lineage that was predominant was the B.1 lineage. If we compare the 2022-23 outbreak with the current ongoing outbreak, so there are 2 to 3 main differences. First of all, the previous outbreak was mainly in a group which was termed as the MSM, men who had sex with men, whereas in the current outbreak, this group, along with children, they are being infected. Secondly, the previous outbreak, as I mentioned, was due to the clade 2B and the lineage B.1. But in this outbreak, clade 1 is the predominant, and we have seen a variant which is termed as the clade 1B. And the cases are primarily reported to this clade in the current outbreak. In the previous outbreak was very unique due to the fact that it was caused, or it spread to countries that were not previously known to be endemic for Mpox. So in the current outbreak, the disease is so far mainly confined to the Central and the West African countries. So these are some of the differences that can be seen between the two outbreaks or the two public health emergencies of international concerns. In this study, we enrolled 20 suspected cases who met the case definition for Mpox, and so lesion swab was taken from these cases, and the viral DNA was extracted using a commercially available kit, and similarly a real-time PCR was performed. We also had a blood sample from 13 patients, so we performed the VZV IgM using a commercially available kit. All the patient samples who had CT value less than 20, so Mpox positive cases with CT value of less than 20, were further subjected to whole genome sequencing using a metagenome approach. So we use the Illumina DNA prep kit. This kit can amplify any viral or bacterial DNA. And this sequencing was performed on the Illumina IC platform. So for downstream analysis, we used FastQC for raw reads quality assessment. Similarly, a number of tools were used for adapter and duplicate removal. And as for assembly and alignment, spades and BWA tool was used. We use the Genius software for consensus generation, for consensus faster generation, and similarly for phylogenetic analysis, a number of tools such as MAFT, MEGA, IQ3, and FIG3 were used. For mutation analysis of Mpox virus, we use the NextClade. So our result shows that among the 20 patients, there were 9 patients who had travel history of Saudi Arabia. And if we look at the first case, so first case was a 41-year-old male who returned from Saudi Arabia. He had fever and lesions on his body. And based on PCR, he tested positive for Mpox, and the CT value was 18. The second case in our study was again a traveler, a male patient who traveled from Saudi Arabia. He had lesions on his body, and his PCR CT value was 36. The third case was a female, 19 years of age. She also traveled from Saudi Arabia, and on return, she had lesions on her body, and she also had lymphadenopathy, and her PCR came out as positive for Mpox, and the CT value was 16. The fourth case was a male patient, 50 years of age, who was a traveler returning from Saudi Arabia, and his PCR CT value was 34. Interestingly, we also, as I mentioned, we tested serum samples for VZV IgM. So we came to find two cases that were positive for VZV IgM. And obviously, these two cases, they were negative for Mpox. Mpox has two clades. They are designated as clade 1 and clade 2. So clade 1 has been shown, so clade 1 cases, they are more severe, and the case fatality of around 10% has been reported for clade 1. For clade 2, the cases are normally less severe, and case fatality of around 1 and less than 1% has been reported. In our study, when we did the phylogenetic analysis, we found that both of our positive cases that had CT value of less than 20, they were identified as clade 2B, and the lineage was the A.2.1. So interestingly, this is a very rare lineage of Mpox virus, and as I mentioned previously, the 2022-23 outbreak was due to the B.1 lineage of the clade 2B. So we found that our two positive cases, they were infected with the A.2.1 lineage. And these cases, they showed high homology with A.2.1 viruses that were reported in Slovenia and UK in 2022. Interestingly, if we look at the available data at the time, so there were around 45 A.2.1 lineage Mpox sequences available on GISID, and most of them, around 38, 38 of these sequences, they were available from India. And not all of them were patients. Some of them were basically, some of the patients were resampled during different time intervals, and the sequencing was done. So interestingly, our sequences, they showed distant homology from the Indian strains. So again, I think this is really important to look and further investigate on the introduction of this A.2.1 and how this virus or this lineage basically evolved. We also managed to do the whole genome sequencing of the VZV. And so there are 5 clades of Varicella-Zoster virus. Clade 1 and 3, they are mostly common and prevalent in Europe and USA. Clade 2 is found in Japan and in China, whereas clade 5 is found in India and East Africa. So based on our phylogenetic analysis, we found that the whole genome of our VZV virus was, it basically clustered into the clade 5, and it showed high homology with strains that were reported from Germany in 2000 and in 2007. We also performed the mutation analysis of our Mpox virus sequences, and we compared it with a reference sequence. And if you see, so Mpox basically is a large, Mpox virus has a large genome around 200 kb in size, and to be precise, it's around 197 kb and has around 200 genes. So we found in our viral genomes, we found 27 mutations, and some of them were deletions, whereas most of them were substitutions. If we can see that there were mutations in three major groups that basically involve the host immune response proteins, the proteins that are involved in host membrane binding, as well as in DNA replication and transcription. Again, the exact role of these mutations on viral immune response or in viral fitness, it needs to be investigated. So, if I conclude, so obviously, laboratory diagnosis is very important. We can see that during the current outbreak, there are majority of the cases they are not diagnosed through laboratory, and this is because due to the lack of testing kits available. So laboratory diagnosis is very crucial when we talk about appropriate responses. Differential diagnosis again is very key. So Mpox has some signs and symptoms that correlate with some of the other diseases. So differential diagnosis again becomes very important for timely management of Mpox positive patients. And secondly, enhance genomic surveillance we recommend, because it's very important to timely identify the clade and the lineage that is circulating. So as I mentioned previously, in 2022-23 outbreak, the clade 2b was the predominant and the lineage was the B.1. However, we found a very rare unique lineage in our study subjects. And also the importance of metagenome. So using metagenome, we can find different co-infections or different viruses infecting the same patient. And then lastly, surveillance and response is also again very important. It's highly important that we continue to monitor the evolution of the virus, because it has implications on diagnostics, on the management of patients, as well as on the response to an outbreak. And lastly, we recommend robust surveillance at point of entries. In our study, all of these cases, they were travelers, they were people who traveled back from Saudi Arabia. So screening suspected cases at the point of entries is a very useful tool in timely identifying these cases and then in their isolation. So this is all. If there are any questions, I have mentioned my email. You can send questions on my email and I'll try to answer that. So thank you so much.